Thanks a lot.
Thank you, Dr. Sab. Thank you very much. So we can start by inviting Ila Bronsky. Are you there, Mohitji?
Yes, I'm here. Thank you very much for the introduction.
Yeah.
And I think you've summarized my presentation, so maybe I should just open the form for questions now.
Yes, please. Yeah.
I'm joking. Well, thank you for this introduction. It's great to be here, and thank you again for making this a forum that I hope will be both a short presentation and also an opportunity for us to interact and get to know each other. And I am going to be looking forward to continuing this relationship in the future.
Yes,
so let me start off by introducing myself. My name is Ilya Brodsky. I am one of the co-founders and currently the CEO of Pine Biotech. What our company does is we have a vision to enhance human health and well-being by enabling biological research and discovery with relevant data, solutions, and support. Our mission is to simplify bioinformatics and advance research through our modular and intuitive multi-omics analysis and integration platform that is powered by both human experience and artificial intelligence. What we offer are training programs, research support, and big data analytics tools and solutions that are designed for research and biomedical discovery by a wide range of users. And our mission is to make it possible to do cutting-edge research and solve big data challenges by developing innovative solutions that are easy to use, providing training to use them effectively, and arrive at sound and reproducible results. So I hope that today in this presentation, I'll be able to offer you a glimpse into some of the types of projects that we deal with, and also encourage you to think about how your expertise in bioinformatics and your interest in data science could be a way to make a difference in people's lives. So beyond myself, our company, just briefly to tell you about the company, we're essentially an extension of a bioinformatics academic research center at the University of Haifa in Israel. And I am privileged to work with a team of an international team that includes a number of people like the founders of the company, Dr. Alfred Tauber, who has a biomedical and biochemical background, and Dr. Leonid Brodsky, who has a collection of publications and expertise in algorithm design and big data analytics, as well as our team here that you're probably already familiar with, Dr. Mohit Mazumdar and Bipsa Biswas, who you have been interacting with. But there are many others that are making this possible, and so I'm grateful to work in this diverse and talented team. So let's talk about bioinformatics for precision medicine. And what I want to focus on today is the aspect of precision and personalization that we'll talk about in the context of clinical applications, but also the clinical ecosystem. So the system is comprised of a number of different areas that all are being disrupted by this availability and accessibility of big data. And the key is bioinformatics, right? So I'm talking to you as those that are interested in bioinformatics, that understand the data science and the data role in biological discovery and application, translational research. And so I'm encouraging you to view this presentation as an opportunity to leverage your skills, both in academia, but also thinking about industry and the need for professionals in this, with these kinds of skills that are able to solve pressing challenges related to human health. So the future of medicine, as was already pointed out, is driven by availability of data, the expansion and standardization of high throughput sequencing that is resulting in medical practice that is both ultra personalized and precise. And this future is really driven by high quality precision data, deep analytical expertise that is required from all participants of this interaction, and particularly at the systems level, it is the ability to integrate a diverse collection of different types of data sets. So let's take a look a little bit at how did we arrive here, and kind of like what is characterizing the situation today. So a little history on genomic data. So about 70 years ago, the structure and function of DNA was discovered, and it took about 50 years from that point in time until we had the Human Genome Project and that the human genome was sequenced. Eventually, the technology became more accessible, and now we have over 100,000 human genomes. And as you can imagine, the cost and the technology driving this change plays a major role in the adoption of next generation sequencing and making it clinically relevant. So let's take a look a little bit at the economics of this data. So this growth in high throughput data is driven by this cheaper, faster, and more efficient sequencing. So the different formats of sequencing today, we call it next generation sequencing or high throughput sequencing. The technology improved so much that from around $100 million in 2001 that it cost to sequence the human genome, it took $3 million to sequence a genome in 2008, and by 2017, the cost of sequencing a genome was approaching the $1,000 mark. Today, that price is even cheaper. So just recently, it was announced that you can sequence the genome for just $100. And this number, this $100 genome, was previously considered to be the breaking point of when genomic sequencing is really going to make a huge impact. So this happened, as you can see, just in 2020, and February 26th, this announcement was made. And so why is that number so important? Well, the economic reason for this number being so important is because your other clinical tests are much more expensive. So in fact, you can see here a few examples: X-ray about $250, computed tomography about $800, nuclear medicine about $600. So you're really putting next generation sequencing in a comparable bracket in terms of a clinical application, which makes it now a component of biomedical data that could be regularly relied on by clinicians and outside of biomedical research only. The significance of that is that the economics of data are hugely important for clinical practice. So medical data, or simply clinical data, is the type of information that clinicians have been relying on for centuries. It's temperature, heart rate, other vitals, and that data is sort of on the surface. That means that it is typically measured quickly, it is cheap to generate, and it is already covered by a standard practice in terms of coverage of insurance. But with the advent of cellular and molecular biology, we've learned how health and disease are driven by what's happening deeper. And not only deeper at the tissue or the internal level, but it's really driven at process that is subcellular. So subcellular processes, the genomic variation, the transcriptomic variation, proteomics, epigenomics, and metagenomics together really drive disease and eventually are manifested in these clinical phenotypes that are observed and are relied on to explore, identify, and treat disease. And so as a result, this is now biomedical data that includes the combination of both clinical and omics data, or the information about the manifestation of disease, as well as the underlying biology that is composed of layers of different types of omics. So as a result of this process, we have an explosion of data, but a lag in the information that this data carries. So with increased reliance on data and much lower costs of generating very detailed and huge data sets at each clinical interaction, we're now faced with this explosion in data availability. But a growing informational lag really complicates the translation of this data to become practical and impactful insights at the point of care by clinicians and benefit to the patient. And so that is what we call the analysis gap. It's the difference between our ability to generate new data and the ability to analyze and interpret it for the benefit of an individual patient. So this gap is a disconnect between lab clinicians, researchers, and clinicians that form the biomedical research ecosystem. And the translation of these insights are really dependent on closing this gap. And this gap, it breaks up the cycle of innovation due to the lack of data-driven research skills among otherwise effective biomedical personnel that is dealing with increasing demands and a changing, unpredictable landscape in terms of more and more complex conditions that we're trying to address. So what types of industries are being disrupted by this process? So in this pandemic, I think it's become very clear how availability of data on its own is not really going to be the solution for treating and intervening in this kind of outbreak. And many other more subtle conditions are really a crisis in the works. And so we can see that all aspects of this biomedical ecosystem, the research, the hospital, and the pharmaceutical industry are affected by this gap between the data that is accessible and cheaper to generate and the ability to extract meaningful insights from this data as a whole, understanding disease and being able to make practical decisions at the patient clinical level. So that brings us back to the topic of this webinar, right? Bioinformatics role, the role of bioinformatics, the role of bioinformaticians, the role of data science skills for all those that are interested in precision medicine. And so this role includes the ability to process big data effectively, structuring that data, organizing that data, making that data possible to work with, the rapid hypothesis testing and analysis that includes complex approaches such as machine learning and various deep knowledge of biostatistical methodology, as well as the ability to integrate, annotate, and make sense of identified patterns. So who should be concerned with these topics and who should have these kinds of skills? The goal, I think, is for these skills to be equally adopted across the whole scale of data generation, the analysis of this data, as well as the clinical application of these data sets by decision makers, right? So the translation, the path from labs where this data is generated to people that analyze and interact with this data, and ultimately to the decision makers that have to look at the data, evaluate it, and make decisions about how to take the next step. And so what we really arrive at is at the challenge of multi-omic integration. So when we talk about multi-omics integration, we're talking about the complex regulation of phenotypes at the subcellular level, where each level of that regulation can be studied at a distinct type of data. And each type of that data is specific, as well as has a specific process of translating that raw data into information that we can later on to use to study the system as a whole. So when we talk about this process of integration, we're really talking about looking at how bioinformatics can help us understand the relationship between the different clinical manifestations of disease, the underlying omics data, and the utilization of this information for diagnostic and therapeutic purposes. So let me pause here and offer you a few examples. As you all know, when we look at a patient, here you can see a mammogram or an X-ray picture of the breast, where the doctors have to look for abnormalities. And once those abnormalities are spotted, you have to make a decision about what to do about this abnormality, right? So here in this example, we can see this white area. This white area could potentially be a tumor. And one of the things that the clinician has to make a decision about is what type of whether this is a malignant growth, a non-malignant growth. And so what are the tools that the clinician has to be able to make decisions about this specific growth? One of the tools is you can start looking for what tissue this particular growth is around or adjacent to. So you can look at, for example, the tumor being close to different types of tissues that are typically present in breast tissue. And based on your ability to identify visually where it is, you can start making some characterization of this growth. However, it's difficult from just an X-ray to know what to do. And so oftentimes people go and extract some tissue, biopsy, and they can now use staining or antibodies that have different colors to identify presence and abundance of particular types of receptors. So when we look at these receptors, a typical way to deal with breast cancer classification is to look for 3 different markers or 3 different proteins on the surface of the cell membrane that allow us to easily understand what type and classify, categorize what type of a cancer this might be. Now, over time, people have studied these types of cancers, but the problem is that just looking at these membrane markers always leaves us in the dark about the processes that are driving the disease, because the disease is not driven by these external receptors. It's really driven at a deeper level at what happens inside the nucleus at the DNA level and the expression of what's in the DNA and how it is different from normal tissue at this particular patient's body, in this particular patient's body. So over time, molecular subtypes have been developed looking at a combination of 50 to 100 different genes that can inform our ability to diagnose in a more precise way what type of a disease this is, how dangerous it is, and inform our decisions about what to do with these biomarkers. So these molecular subtypes that are characterized by molecular biomarkers are essentially collections of genes, and by looking at sequencing data, extracting those genes, we can now characterize different types of breast cancer. So here we have kind of the evolution of these breast cancer classification, which happened over time. And over time, we've started understanding that even this ability to characterize patients and put them into different groups is oftentimes insufficient in terms of accuracy of diagnosis, prognosis, and identification of the best response to treatment. So here you can see an example of kind of an evolution in the different types of panels that are used to classify and categorize these different types of patient samples. And the ability to really understand how we can both look at the association of clinical data about these types of cancers and functional classification that allows us to spot early on some processes that are important for disease progression, metastasis, cell adhesion, different kind of classical characteristics of cancer. So how are these biomarkers used? Well, these biomarkers could be used to diagnose, like we just looked at before. It could be, they could be used for predicting survival. For example, both of these panels, PAM50 and Oncotype DX, both of them consisting of just tens of different molecular biomarkers, have been shown over the period of 10 times in clinic, or 10 years in clinical trials, have been shown to be associated with high, medium, or low rate of recurrence and rate of recurrence and ability to predict the outcome. And so this is an important tool in the toolkit of a clinician to be able to make important decisions about treatment. When we talk about treatment, the selection of treatment is really driven by a balance of cost benefit to the patient. Oftentimes, the treatments that you have available for cancer are highly toxic to all the other normal cells in the body. And so being able to predict whether a patient has 6 months or 10 years to live or is likely to live as a result of this particular cancer might impact significantly the choice of treatment that the clinician will make. Importantly, however, as it was noted in the very beginning, we are now entering a new era of targeted therapy. And in cancer, this targeted therapy is driven by not only identification of level of risk, but also identification of specific features or specific players in what's going on inside this tumor. So identifying not only prognostic markers, but actual targets, we can now design treatments that can interfere with a molecular process and stop different aspects of cancer characteristics. For example, one of these famous drugs is Herceptin. Herceptin is an antibody immunotherapy that blocks a receptor on the surface of the cell. It blocks and prevents it from signaling to the nucleus on cell proliferation, which results in tumor growth. So we can see how this ability to identify what happens at the subcellular level and not only at the membrane level now helps us understand how this could be treated in a more effective way. So for example, immunotherapy has already transformed cancer care. And many of such precision drugs are the result of biomedical research studies of the tumor, and not only the tumor itself, but also the microenvironment. Mapping the changes that characterize tumors and understanding the biological pathways inside tumor cells and signaling between tumors and their microenvironment have led to innovative interventions that are blocking, suppressing, and enabling existing mechanisms of action to mitigate the effects of uncontrolled proliferation and metastasis. So this is a drug called Optivo. This drug was approved for medical use in the United States in 2014, so just 6 years ago. It's on the World Health Organization's list of essential medicines, the safest and most effective medicines needed in a health system. Optivo is a medication used to treat a number of types of cancer, including melanoma, lung cancer, renal cell carcinoma, Hodgkin lymphoma, head and neck cancer, colon cancer, and liver cancer. And these benefits of such targeted drugs really require for analysis of data that comes from these different tissues to discover biomarkers that will allow the clinician to guide treatment selection and evaluate the effectiveness of treatment along the way, really placing data analysis at the center of such precision treatments. So there are a few other examples like Optivo. For example, nivolumab is used as a first-line treatment, right? So Optivo comes in a variety of different packages, and nivolumab is one of these other names for it. And it's really useful for metastatic cancers, and which is very important. And the mechanism of action is actually dependent on specific mutations in different genes. So we really are looking at a few examples of how receptor-driven, so gene expression and protein expression level, mutation level data, all of these types of information are really useful for a clinician to make a decision. But here we're talking about some advanced cancers or advanced medical conditions, like a tumor that is proliferating and already growing. What about early on? Well, increasingly, it has become clear that disease like cancer is not only caused by genetic mutations and aberrations in gene expression of the host machinery. Really, what's become apparent is that many diseases are driven by variation at viral pathogenesis that takes a long time to manifest itself as a disease. It's also driven by changes in the microbiome and composition of the microbiome. And today we know that some of these major players like the Epstein-Barr virus, the human papillomavirus, and hepatitis B and C are major pathogens that over time can cause significant damage and inflammation to healthy tissue, resulting many times in hundreds of thousands of cases of cancer per year globally. So this ability to identify at an early stage a presence of a particular infection and pathogen in the patient is critical for our ability to intervene earlier on and also treat not only the manifestation of disease, but also the agents that cause rise to that disease. So today, when we talk about cancers, the need for cancer diagnostics, right? If we talk about what's the impact of bioinformatics on cancer treatment, you can see that actually applicable in all areas of cancer management, which is diagnostics, so accurate diagnosis using molecular profiles, understanding how different types of genes, mutations, epigenetic variation are playing a role in accurate diagnostics on an individualized level. It's discovery of targets, like we just talked about, of targeted therapy that is going to impact particular pathways and mechanisms, as well as identify what is going on, what is driving that kind of aberration, predicting risk, prognosis, so identification of specific features that allow us today to look at what will happen over the course of the next several years of a patient's life to understand how the disease will progress and what are some likely outcomes and risks. At the same time, we're talking about regular application of data analysis to really understand responses to treatment. So pharmacogenomic treatments, markers that allow us to track the effectiveness as well as the potential dangers that new treatments might implement at the patient level. And finally, stratification. So one of the challenges today is to accumulate knowledge about every single one of these processes, making it possible for us to understand how a particular person or the markers that we have discovered characterize the biology of this individual. So many times we have to rely on data alone to understand how patients could be organized by groups, how they could be associated with a group that is being studied in clinical trials, how could it be associated with experimental data where we don't have data on patients, but we have a lot of data at the cell line and the animal model level. So really, there's a huge need in regular processing of data, as well as integration of this type of data across a variety of different domains. So you might have heard that in 2014, there was an outbreak of Ebola in West Africa. This was one of the first pandemics or epidemics, because it was localized, that gave rise to a huge variety of clinically applicable next generation sequencing data. So the Ebola outbreak of 2014-2016 really marks the beginning of clinical use to study epidemiological outbreaks of infectious diseases. Today, we use this kind of information to understand diseases at a very detailed level. And so the same type of information that we discussed for cancer understanding and treatment are now being applied to study these infectious diseases. And more than at any time previously, we can appreciate the impact of sequencing on SARS-CoV-2 or COVID-19, right? So today, when we talk about sequencing data, it's not even something that you have to explain the utility of. Everybody understands that the understanding of the genome and understanding of how that genome changes over time as it spreads through between different hosts like animals and people, as it spreads between different populations in different regions around the world. We know that this kind of information at the genomic level really helps us study the disease to generate information that is critical for the next steps as we try to intervene. So when we talk about these kinds of findings, we're really talking about a variety of applications of molecular subgenomic data that allows us to understand both the epidemiology as well as the application of these findings to novel treatments and an understanding of the basic biology. So you've already probably seen how very quickly from just this year, right, starting in December 2019 and all the way until today, thousands and thousands of genomes from patients throughout the whole world were generated. Many different collections were prepared. In fact, you now have a whole list of sources and databases that you can research and explore genomic data on COVID-19. And what's important about the growing availability of this data is that it is becoming more and more complicated and at the same time informative. So what we can see here is the ability to track mutations worldwide, understand how this type of mutation data or genomic data can explain variability of disease, and we can study how host factors, right, the response of different individuals, young, old, of different genders and backgrounds with other types of comorbidities, helps us to translate that data into models that actually inform not only an individual treatment or individual interaction with a patient, but actually at a global scale, right? We now know that this kind of genomic data is impacting decisions by governments, impacting decisions by health systems. It's helping health systems develop novel diagnostic tools. And we are looking at a particularly fascinating, in my opinion, trend where we are discovering how drugs could be repurposed as well as modified to address specific challenges like what we're seeing with COVID-19. So here you can see a couple of publications that were focused on the integration of molecular information like the protein subnetwork associated, so COVID-19 and human associated host protein subnetwork, the interaction between the viral proteins and the human proteins, and an associated network of protein drug target network. And based on that information, you can actually find important clicks of that system, of that network, to target a particular interaction with an already known drug. And so really this is called network-based drug repurposing, where you can analyze networks of drug interactions, protein interactions, host pathogen interactions, and identify some effective breaking points that might be accessible that could be further studied. However, not only is this driven by more kind of like not knowledge driven, it's really data driven research, because ultimately the proposed approach then, what you can see here on the right, is the approach of high throughput sequencing, both in cell line experiments where you can rank compounds and rank thereby efficacy, as well as testing of those compounds on animals, animal models of human disease. And in all of these different aspects, from cell line experiments to animal models to then application to human population, all of this is driven by this ability to integrate data and track similar patterns across the different domains so that we can inform decisions at every step of the way. So, to summarize, how is such high throughput data transforming the clinical and pharmaceutical industry? When we talk about the applications of bioinformatics to biomedical research, we're really talking about clinical uses such as patient stratification, disease classification and diagnostics, identification of potential responders to therapies, as well as the study of the mechanisms of disease onset and progression at a much deeper level. And at the pharmaceutical level, we're really talking about discovery of biomarkers, detection of toxicity at the in vitro stage, so before it reaches the clinical application, analysis of molecular mechanisms of drug action or target discovery, as well as drug repurposing and repositioning. The amazing thing that we're seeing today is that the wider biomedical community is becoming much, much more open, because the need for data analysis skills, tools, and an understanding of the important applications of these methods is becoming more of a need than privilege, right? So previously, a lot of this kind of research was very diverse and separated and required highly trained experts that could participate in such studies. Today, the need is much bigger, and so it is becoming much, much more open and ripe for disruption. The key for closing the gap between the ability to cheaply generate data and making sense of how to use this data are really tools, skills, and a deep understanding of the application. So what we do is provide such training. Sorry, let me go back here. What we do, what our company does to address this gap is we provide a collection of tools and training to discover the important questions, such as the ones that I have described today in this presentation, and plan for an environment, an ecosystem, where one can flourish and develop independent projects using this type of data and showing the impact on a particular domain. So our OmicsLogic training programs, I'll briefly describe those, are really designed of several components. And I think it's important to understand how they could be helpful, both in the long term for impact on healthcare, but both from an educational and academic perspective, how they can translate a program that deals with bioinformatics or biomedical research to make them more effective at addressing these problems at the industry level. So what we offer are interactive sessions, self-guided materials that are packaged under these different collections, as well as practical assignments. And these elements together provide a resource that can supplement any kind of an academic program to really make a translational impact on how these types of materials and data sets could be applied to solve real challenges. So what we hope to do as a result of doing this for the past couple of years is to provide a clear perspective on bioinformatics, machine learning, and artificial intelligence in research, clinical, and industry applications. The way we do this, as I mentioned, is by providing access to tools and enabling infrastructure to deal with big data. And I'll talk about this in a second. We also provide the logic of analysis in application to real data sets. So we don't just deal with hypotheticals, we actually take the examples like what we've talked about up until now. Different types of cancers, different types of treatments, different types of risks and strategies, different clinical interactions, as well as epidemiological genomic data. We also provide practical applications to a variety of projects. So our goal is to prepare a person and provide them with a springboard to jump into a real situation that might have an impact, that there is some area of that data set that is unexplored and you can really try to make a difference. A lot of these different types of projects really deal with the ability to integrate multiple factors that cause disease and drive disease progression. That includes cellular omics, so a variety of different types of omics at the cellular and subcellular level, the microbiome and the virome, as well as the integration with clinical or phenotypic data. And as you can imagine, a lot of these different associations or correlations are nonlinear, complex, and require a lot of manipulation and playing around with to really arrive at discovery. If you were to try and do that on your own system using only the resources that are available to you, you will quickly run into the problem of time. Every single step of the way to process this data, to try and integrate different parameters of the data that you found requires a lot of time. And so a critical part of what we provide is facilitating infrastructure to speed up that process. In fact, we've developed a multi-omics integration platform. It's a cloud-based platform that allows us to streamline the process of generating structured data and mining it for associations. What types of methods does this platform include? As I mentioned, this is a multi-omics data, so it really has different types of sections like next generation sequencing data analysis, mass spec data, structural biology data, as well as a whole section on integration and modeling. Within each section, to speed up the process of understanding the logic of analysis, we provide demo pipelines that you can try and run with a demo data set to really understand what are the inputs and outputs and the logic of how to build a pipeline that's going to be effective. There's also a more growing interest in machine learning and artificial intelligence. And so we believe that every bioinformatician today needs to be familiar with some of these technologies, how they operate, what are the different settings for using them effectively, but also how to apply them to solve particular problems. And finally, we provide tracking and resource management that allows you to easily transition between an educational situation to an application. So actually there's a transition from basic to project-based education to then application this to real research projects. So in fact, we have collaborations with many different universities that are focused on application of many of these different processing tools and analysis tools to translational research projects. Many of these projects, many of these types of processing data pipelines are really designed to provide a combination of color-coded maps, explained methods that you can really dive deeper into what each method does and understand what are the parameters that are important for running that properly. Outputs that are both visual and technical that you can utilize in future downstream analysis, and importantly, a reproducible workflow. That reproducible workflow allows us to provide high quality technical support and understand what are the challenges that participants face, as well as understand for a user of these types of tools to be able to share your analysis in a very simple way for other people to benefit from. In addition to this platform, we provide training resources that are really packaged course materials that could be supplemented for any other type of bioinformatics training that explain this kind of logic at a very user-friendly level, as well as on a more technical level, going behind the scenes and understanding what are some of the coding skills that are required to run these types of analyses in both R and Python. Moreover, and I think very importantly, is that this type of education is really project driven. And I think that if we are trying to make a difference in the way that precision medicine is improved by application of bioinformatics to high throughput data sets, it's important to understand that these types of skills are applied at all level of biomedical research and all the way to clinical applications. And so we've really worked hard on interacting with our partners from academic centers and clinical centers to source projects of high impact that could explain some of these paradigms that we've discussed in the workshop and how bioinformatics could be applied to see them in a real project. So when I say project, I really mean taking a publication from a high impact journal, understanding what type of data was used in a particular study and with what types of methods it was analyzed, and looking at that published and digested example. It's not a textbook example, it's an example from a real study, and we try to keep them up to date as much as possible. You can start understanding how the methods and the data combined together could actually yield a novel idea, which is independent and turns into a project of your own. So what types of projects am I talking about? One example of a project is exactly like what we discussed with bioinformatics for precision medicine or for breast cancer classification and selecting biomarkers to allow for precision treatment of breast cancer. So in this example, you can just look at the abstract and immediately understand what the authors were trying to do. What they did is evaluate the benefit of different types of molecular data, 7 different data types, to associate with different types of clinical trial level drugs, as well as approved on the market drugs, to understand how you could develop an analysis workflow to select the optimal treatment for a particular patient. But this is just one example. So in fact, we have a variety of projects. These projects range from cancer to neuroscience to agriculture to different types of infectious disease studies that I think all illustrate how important bioinformatics can be in this process of changing healthcare the way that we know it today, making it more impactful at an individual level, making it more precise and less toxic with less side effects, and ultimately driving the treatment of these complex diseases to allow for an effective treatment of diseases that have not been able to been treated before, right? So many diseases that we know of today, including COVID-19, really do not have an effective strategy to be prevented or treated at this moment. And bioinformatics is, I think, very key to discovery and application of these treatments. So what kind of impact have we made so far? I think that, you know, it is easy for us to kind of look back at the last year and know that this impact is really providing results. So what I've seen is thousands of students, including high school students, undergraduates, graduate, and even postgraduate students, including postdocs and researchers, that have seen how these programs help them address challenges in research, discover new information that was previously inaccessible to them, and ultimately make a difference. So our programs include a variety of different types of basic training, but also because I know that many of you are graduate students and postdocs, I did want to mention that we have a research fellowship that our company supports, which is a program specifically designed for those that are interested to apply bioinformatics to some of these cutting edge precision medicine related projects. So this research fellowship program includes really experience in application of bioinformatics to big high throughput omics data, where we help find and select an appropriate project and data set. We help master the tools that are cloud-based and effective at discovering specific patterns in different types of omics data. Understanding of how that different types of omics data play a role in different types of biomedical studies. And ultimately, we also help develop a set of kind of validated projects or skill sets that you can later on take with you when you apply it to your research or industry applications when you might be participating in the industry research with these bioinformatics tools. So at this point, maybe we can share the link just for those that are interested in some of these programs. We have this research fellowship program that I invite you to look at. And also, if you have any questions, I would be happy to answer any questions that you might have at this time. Thank you. And Bipsa, posted some of those links in the chat for those of you that are interested to take a look.
Hello.
Yes.
Yes, I'm myself Dr. Ashok. So I need to ask you one thing. Oh, if you really are looking for some special skill before we enter to this research fellowship, as such we talk about, what's the prerequisite if you want to apply?
So there's really two options for the research fellowship. One research fellowship option is a 3-month accelerated program for those that are already familiar with some basic bioinformatics. So they are familiar with different types of omics data. They are familiar with processing tools for omics data. They're really ready to start a research project. So that typically lasts about 3 months, and the goal is to make an impact. So driven by a poster presentation at the end of that process or a research publication. And we have a community of people that you can collaborate with in that project. The other option is for those that do not have those skills. So they really are interested, they maybe know the biology or some clinical application that they see themselves driven by, but they don't have a lot of coding skills, they don't have the biostatistics skills, they don't have the bioinformatics skills. So that program is 6 months, and that includes training, which includes access to all of our programs that are ongoing, so you can explore different tools and different project examples. And then at the end of that process, you still have the 3 months to develop an independent project.
So if you talk about this project fellowship, so are we getting some sort of a degree or certification after completing the product, and the output of the project will be a proprietary product of a company?
No. The output of the program is a project that you take with you. So the objective of this is to develop a research presentation, which could be a poster or a publication or a manuscript. So really that is the outcome, and it's not for undergraduates, so it's not certificate based, it's really based on that project outcome, right? So our goal is to what we do to support that is also
one thing more. If we enrolled in this particular 6-month project, so the tools we'll be learning, so it can be used in our online or open source system, or you have to be dependent on all your services you are providing?
No, you can use it. We provide you with cloud access to our tools. If you're interested to implement those tools in other infrastructure, you're welcome to do that, and we can help you set that up as well.
Okay. Thank you.
What is the cost of the internship program for research fellowship? There are 2 different modes here. Like I said, 3 months and 6 months. So if you're interested in that, you can probably email to marketing at Pine Bio, and all the details could be sent to you by email. For those of you that are interested in doing more with your bioinformatics and becoming a part of a growing bioinformatics community that is applying these kinds of tools to a variety of different challenges, we also welcome you to join some of our community on social media. We have a group on LinkedIn with over 500 professionals from these different pharmaceutical and clinical applications of bioinformatics, as well as a group of over 5,000 people on Facebook that you're welcome to join as well. Okay, what would be the difference between these 2 internships? Really, it's the length, and that's dependent on your background. How much background do you have in bioinformatics? That's for Aruj. Nupur, can it be pursued during postgraduate course? Yes, so we have a lot of people that are postdocs, PhD students, and even researchers that work together as group projects. Many times you have a faculty member that is driving the research project and would like to rely on some student support or other type of support. So yeah, there's opportunities for those as well. If you have a master's in bioinformatics, yes, you're definitely the more applicable is the 3 months internship. And Beeps posted there a link to our LinkedIn group. So you're welcome to join that group. We hope to use this group to make introductions. There's a lot of job postings in both academia and industry. if you already know bioinformatics and have something to show. And what we hope also as a result of this research fellowship program is to show how you can improve your CV or your way that you present what you know about bioinformatics when you apply for these kinds of positions. The eligibility is really, like I said, there's two different types of programs, 3 months and 6 months. The 3 months program, if you want to get into that program, you have to already know bioinformatics, which includes either a certificate that you already have for bioinformatics training program, such as the ones that we provide or many other universities provide, or a degree in bioinformatics. If you do not have a degree or a certificate program, then you can still get into the 6 months fellowship, and that will give you both the training and the ability to then develop a project of your own. Okay. Well, again, it was great for me to be able to speak with you today. I'm excited that we will be able to form a longer-term relationship with Punjab University and explore a lot of these topics in greater detail. For sure, we've done a lot of collaborations that lead to research projects at the end at both individual student level, but also at the program and university level. So that's really the hope for our side. Okay, so a question from Aruj. You have a master's in biotechnology, but your research was related to bioinformatics. Again, I would recommend emailing Bibsa with your current CV, or she also has a link. Bibsa, maybe you could post a link again. Once you submit the form, we will be able to better address the needs of the program for your individual case. Okay, there's a, okay, let's take a look if there's a problem with the link. Okay, so this is the link. You can see here some information about this. So it seems to be working. If you're having trouble, please again email Bibsa and we'll be able to send you that information by email. All right, great. Well, I see many of you are now joining the LinkedIn group, so it's great to have you with us here, and you can explore some of the members and have a lot of experience in different areas of industry and academia. And okay, will we share the sessions? Okay, so maybe just in closing, I also have another meeting here that is coming up, but in closing, I would like to share with you that we do have 2 other upcoming programs. One is focused on metagenomics, or a program on microbiome. That is for people that do have some familiarity with bioinformatics. We'll go through some of the typical tools and some exciting project examples that are related to therapeutics and are related to different diseases. So I welcome you to take a look at those, as well as a program on transcriptomic data analysis. So it's real training on RNA-seq, including bulk RNA-seq and single-cell RNA-seq. So you're welcome to join the transcriptomics or explore the transcriptomics program as well. Those will be starting soon. We do have a growing presence in different universities in India, and we hope to develop more. So we also are hoping to develop joint certificate programs because there's really a huge demand for these types of skills internationally, and especially in the era of COVID, as you know, you can definitely work from home, and so a lot of companies and positions are looking for that kind of support. Dr. Kumar, thank you again for...
Yes, dear Eliaji, this interaction was very interesting, and we get a lot of opportunity to ask and interact. So hopefully we'll be interacting with you more. And thank you very much for your presentation. And then I think participants are much more clear what is possible, and they'll be interacting with you more directly on email. In conclusion, we can say, Mohit want to say anything, then we can conclude.
Yes, Dr. Ashok, thank you so much for this webinar. And I think it was really useful in terms of understanding the application of this technology. And I'm really happy that all the participants were able to join and ask questions. And we will be sharing the video of this session to everyone so that they can review it later as well. So thank you again, everyone.
Okay, Ji. Okay, then we can say conclude. So we can declare this meeting and end. Is that okay?
Okay. Great. Yeah. Thank you very much, and looking forward to continuing this interaction.
Okay. Take care. Good night. Bye bye.
Thank you. Good night. Thank you.